Hello, it's Felice Tirone speaking from the National Research Council of Monterotondo, Italy. My laboratory has been working since many years on the development of the cerebellum and its pathology, the pediatric tumor medulloblastoma. We will summarize our work and the mouse models generated. Cerebellum development continues after birth in human and mouse by massive proliferation of granule precursor cells at the surface of the cerebellum under the proliferative stimulus of sonic edger. Precursor cells then migrate inwardly and differentiate, as you can see in the cartoon. This massive proliferation creates susceptibility to cancer. In fact, precursor cell may become neoplastic, developing medulloblastoma, as you see in the GFP positive image in green. In children, the medulloblastoma radio and chemotherapy leave permanent cognitive loss, so there is need for new therapies. We studied two genes that we demonstrated to control the cell proliferation and differentiation in cerebellum. BTG1 inhibits the proliferation of granule precursor cells, while TIS21 is required for their migration inward and differentiation, as you can see from the in vivo images and graph. As we had previously found that TIS21 is a medulloblastoma suppressor, we generated a new mouse model by crossing patched 1 heterozygous mice, which develop spontaneously medulloblastoma, with TIS21 knockout mice. We found that the ablation of TIS21 highly enhances from 25 to 80% graph, you see graph on the left, the incidence of medulloblastoma spontaneously occurring in patched 1 heterozygous mice. This occurs because mutated precursor cells do not migrate internally and differentiate, but rather remain longer in the proliferative area at the cerebellar surface, becoming target of transforming insults. We looked for the genes responsible for this migration defective phenotype by microarray analysis of cerebellar precursor cells. We found 22 medulloblastoma-specific genes involved in cell migration, and among them we identified the chemokine CXCL3, which, being able to regulate the migration of cerebellar precursor cells, was potentially responsible for the increase of medulloblastoma frequency. And in fact, we observed that the delivery of CXCL3 in the cerebellum for 1 month with osmotic mini-pumps stimulates the migration and then the differentiation of preneoplastic cerebellar precursor cells and prevents completely the growth of medulloblastoma lesions. We are continuing these preclinical studies in order to start experimentation in humans. We also performed a gene therapy preclinical trial testing the effect on the growth of a mouse allograft model by an adenovirus expressing TIS21. We observed a significant inhibition of growth of medulloblastoma nodules by the TIS21 expressing adenovirus. Finally, we generated a mouse model of spontaneous medulloblastoma carrying a knockout of the gene BTG1, which controls the proliferation of cerebellar precursors. Surprisingly, we did not find any increase of medulloblastoma frequency, but we observed a large increase of tumor stem cells, which are responsible for long-term relapse. In conclusion, we aim to continue the preclinical trials with CXCL3 and TIS21, and to exploit the BTG1 medulloblastoma mouse model showing increase of tumor stem cells in order to study the medulloblastoma relapse.